All right, so previously what we discussed is this strand and complementary strand turning on itself in a right-hand turn to form this helix. Really properly, I should call this a double helix. And this double helix is basically turning on itself to give me this corkscrew shape, and this corkscrew shape is what we associate to the DNA strand itself. Okay, so we talked about these major and minor grooves and the importance of those grooves and how to determine them. And we also talked about Watson and Crick and Franklin's discovery of the structure, the 3D shape at least, of DNA itself. So now what we want to talk about is maybe some other things that come along with DNA that's more in the biology field than it is in the chemistry field. Notice so far this lecture maybe feels a little bit different to you because we've not really seen structure, we've not really seen nomenclature, we've not really played a focus on reactions that can go on. This is just really kind of information on a biological perspective, a macromolecule perspective, instead of the actual chemistries that are associated with them. So, one of the first things that I want you to realize is that this diameter may be tiny, but don't be surprised, this molecule is very long. So, it could actually become 6 foot long or even longer as far as a molecule goes. Could you imagine a molecule that is 6 feet long? Well, that's DNA. And again, that's because DNA is made up of a billions of base pairs. I think the current count is 3.1 billion base pairs that DNA has as far as the ladders go. So those AT pairs and those GC pairs, 3.1 billion of them in a normal kind of human length DNA chain. Okay, so we need to talk about how this 6 foot long thing is packed inside of your cells because this is quite an accomplishment, really. I mean, sometimes the small and large intestine impress me, knowing that these things are feet long and they are crammed inside of me, basically, and they are put into a position where everything fits. The same thing has to happen to DNA. We have these huge, long pieces of DNA, 6 foot long or longer, and this DNA has to be crammed inside of a little water balloon known as a cell. And not only that, but they have to be crammed in more compartments inside of that water balloon that are even smaller than a cell. So this kind of blows my mind for a minute. How can something this large come from something so tiny, right? So we're going to have to start talking about packing. How do these things basically pack? And the very first term that we like to throw out is something called a histone. All right, so we need to talk about what a histone is. So imagine a histone as kind of a bead or a particle in a way. And then what happens is that this straggly little DNA strand actually takes a look at this histone and begins to wrap around it very compactly. So around and around and around and around and around. And then maybe another histone could form, and around and around and around and around and around. And then another one goes, and then it's around and around and around and around and around. That is what we call a histone, and you're going to see that in the actual PowerPoint itself. So when we talk about histones, we're talking about basically packing DNA into a nucleus of a cell. And the first route to do this is with the histone. So what I want to show you is a video that basically helps put all of these pieces together for a minute. So let's take a minute and let's just pull up this video and let me kind of talk you through that video as well. Okay, so here's a video that's going to basically explain what a histone is and how that histone basically relates to these things called chromosomes that you've heard about in biology class before if you've taken one. So you probably won't be able to hear the sound. Maybe you will, maybe you won't. But if you don't, the link is in the PowerPoint, and you're more than welcome to click on that link in the PowerPoint and open this up on your own computer and take a look and a listen at this video that begins to talk about histones.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell.
Okay, so what you're seeing here is a picture of a DNA molecule. And this DNA molecule is the coil. So you can see the coil wrapping around and around and around and around and around. And we get this nice kind of right-hand turn. We see the base pairs in the center, which are here, and then the sugar and phosphate backbones are wrapping around the outside. So that's the first thing that you need to realize. This is a DNA molecule, and it's substantially long.
The process starts when DNA is wrapped around special protein molecules called histones.
Okay, so first off, these histones that we're talking about are proteins. So those proteins are made up of amino acids. They're polymers of the amino acids, right? They have a primary, secondary, tertiary, quaternary structure. So that's what these blue kind of things are. These are our histones. And what you're seeing is that the DNA is beginning to wrap around these histones as they are introduced.
The combined loop of DNA and protein is called a nucleosome.
Now you're seeing these multiple histones basically coming together, and they will form an even more compact structure for you.
Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then...
So chromatin now has been formed. It's these multiple histones that's now been packed together even more. So we've had DNA packed around the histones packing around each other. That is called chromatin. And now we're going to see something else that happens.
Looped and coiled yet again.
So now the chromatin is actually going to be looped and packed together even more. So it keeps going over and over and over. It's like those little German toys, those little German dolls. You open up one and there's another one on the inside, and you open that one up and there's another one on the inside, and you open that one up and there's another one on the inside, and it keeps going on and on and on and on. That's kind of what's happening here. So there's all the chromatin that's been wrapped around on itself,
leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not...
And these are chromosomes, of course. So if you can't hear the audio, these are chromosomes inside of a cell, and the cell is getting ready to split, and this is actually being seen with a microscope. So that's how your DNA is packaged. That's how the DNA twists and turns on itself. That's how a chromosome is formed. And now that's what you're seeing here. The chromosomes are being split.
Not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated.
So here you're seeing a split of the cell. The cell now splits into two, each with chromosome in it, each with the DNA in it. Okay, so that's where I'll stop this portion of the video. This video will continue, and the next portion of this video is going to be something called DNA replication. So we've talked about the 3D shape of DNA. We've talked about how DNA is now packaged inside of your cell and how that packaging leads into what we call a chromosome. And then now in the next portion of the lecture, we're going to talk about how the DNA actually replicates itself and passes information along from one thing to the next. So that's what we're going to be talking about upcoming. Again, this is more biology than it is chemistry. It might be a nice change for you. It might be a breath of fresh air. It might be a part of science that you be like, well, this is not too bad. I kind of like what I'm doing here. And if so, then biotechnology might be a field for you as well. So this is kind of what they do in biotech. They do a lot with DNA. They do a lot with protein. They do a lot with these antibodies. So maybe you found a new trade. Where can you go and have a biotech degree? Well, many colleges in the state have one, so the only thing you got to do is search for it. I'm sure that you'll be able to find it.